Eat a meal at 11 at night and your pancreas will handle it. It has no choice. It will release insulin, blood glucose will come back down, and by the time you get a standard blood panel done, every number on it will look completely fine. But the organ that just did that work wasn't built to do it at that hour, and it lets you know, not with pain, not with anything you'd actually feel, but with a molecular signature that surfaces decades later as insulin resistance, stubborn weight gain, and roughly double the risk of type 2 diabetes in shift workers who eat on exactly this kind of schedule. Your pancreas isn't just a chemical plant that reacts to sugar showing up in the blood, it's a clock. It runs its own genome-encoded timer inside the beta cells, independent of everything else, and when you eat against that timer, the organ still does the job, just worse than it should. Here's the part almost nobody knows. Every beta cell in your pancreas is carrying the same molecular machinery that runs inside the neurons of your brain's master clock. The genes are called clock and BMAL1, and together they produce proteins that pair up and switch on a second set of genes purr and cry, on a cycle that lands close to 24 hours. Purr and cry then accumulate, shut clock and BMAL1 back off, degrade, and the whole loop starts over. This feedback loop was first worked out in the suprachiasmatic nucleus, the cluster of roughly 20,000 neurons in the hypothalamus that takes light signals straight from the retina. But in 2010, Biljana Marcheva and her team at Northwestern published something in Nature that changed how people thought about the pancreas entirely. They deleted BMAL1 specifically inside mouse pancreatic beta cells. Nowhere near the brain, the master clock left completely intact, and the mice developed diabetes anyway. Impaired glucose tolerance, insulin secretion that didn't work properly, a malfunctioning organ caused by breaking one clock gene inside that organ with zero damage anywhere upstream in the brain. Sits with that for a second because it's already strange enough on its own. Your pancreas doesn't just respond to glucose. It has something close to an opinion about what time it is, generated locally, with no input from your eyes, no input from daylight. It expects food during certain windows, and it prepares for that in advance. Synthesizing insulin, priming secretory granules, ramping up the enzymes involved in glucose metabolism, all before a single bite arrives. Eat inside that window, and you're walking into a factory that's already open, warmed up, fully staffed. Eat outside it, and you're asking a night shift running with a skeleton crew to somehow absorb rush hour volume. And there's a second mechanism running at the exact same time, and it comes from a direction most people wouldn't expect. Melatonin. You probably know melatonin as the hormone that climbs in the dark and makes you sleepy. What's far less known is that melatonin also directly suppresses insulin secretion. Beta cells have a receptor on their surface called mTNR1B, and when melatonin binds to it, it triggers a cascade through G-protein coupling that lowers cyclic AMP inside the cell. And cyclic AMP happens to be one of the main signals telling a beta cell to release insulin in the first place. Less cyclic AMP, less insulin, on purpose. By design, this isn't a glitch. Nighttime is when your body deliberately dials insulin sensitivity down, because for almost the entirety of human evolutionary history, food simply didn't show up at night. So there was no reason to keep that machinery running full tilt while you slept. So picture the collision. It's 10:40 at night. Melatonin is climbing, doing exactly what 400 million years of vertebrate physiology built it to do, telling the pancreas to stand down. And a plate of food shows up anyway. The beta cells are now caught between two competing instructions. The local BMAL1 clock machinery, which by this hour has already started quieting insulin gene transcription for the night, and a fresh spike of glucose that demands an immediate large insulin response. The result, when researchers actually measured it in humans, is not subtle. Marta Garolet's team at the University of Murcia, publishing in the International Journal of Obesity, followed more than 400 people through a weight loss program and found that late eaters, people eating their main meal after 3 p.m. relative to their own circadian timing, lost significantly less weight than early eaters. Same calories, same macronutrients, same exercise, same sleep duration. The only thing that differed was clock time. In a more tightly controlled setup, Frank Shear's group at Brigham and Women's Hospital in Boston ran participants through a forced desynchrony protocol. Identical meals, eaten at different circadian phases, under dim light, with zero external time cues. and found that eating during the biological night raised post-meal glucose by roughly 17% and measurably dropped insulin sensitivity in the very same person eating the very same food. There's a mechanical detail worth pausing on here, because it's what actually explains the spike rather than just a delay. A healthy beta cell responding to a daytime meal releases insulin in 2 phases. A fast first phase within minutes, pulling from a preloaded reserve of insulin-containing granules already docked at the cell membrane, followed by a slower second phase as fresh insulin gets synthesized. That first phase reserve is itself under circadian control. The priming of those docked granules is one of the processes BML1 and clock directly regulate, building up across the biological day and thinning out toward biological night. So a meal at 7 in the evening and the identical meal at 11 at night aren't being met by the same cell in 2 different moods. They're being met by 2 different physical inventories of preloaded insulin. Blunt that first phase reserve, and glucose lingers in the blood longer before the slower second phase can catch up, which lines up almost exactly with what Shear's team measured in Boston. Now, the evidence calibration, because these mechanisms don't all sit at the same level of certainty. The existence of a local pancreatic clock, and its ability to cause diabetes on its own when broken, is well replicated at this point. It started with the Northwestern mouse work and has since been confirmed in human islet tissue by multiple labs, including Charna Dibner's group in Geneva, who pulled human beta cells out of the body entirely and watched them keep ticking on their own rhythm for days. The melatonin suppression pathway through MTNR1B is also solidly established at the biochemical level. What's less settled is how much of late eating's effect on human weight and glucose comes specifically from this mechanism versus other clock-disrupting habits that tend to travel alongside late eating: less sleep, more artificial light at night, cortisol running out of rhythm. Shear and Gerolit's controlled work isolates meal timing as its own variable, which is strong evidence, but the field is still sorting out how much weight each contributing pathway actually carries. The physics and the biochemistry both point in one clear direction. The exact size of the effect, out in an uncontrolled real life outside a sleep lab, is still being pinned down. There's a third layer worth naming here, because it's what ties the whole thing together. The pancreatic clock isn't running in isolation. It takes its cues from the same source your gut and liver clocks do. And the strongest signal that entrains all of these peripheral clocks turns out not to be light, which mostly governs the brain's master clock, but food itself. Satchidananda Panda at the Salk Institute showed this by restricting mice to an 8-hour eating window without changing a single calorie of what they ate. The mice eating within that window stayed lean. Genetically identical mice, given the same food spread across 16 hours, became obese and glucose intolerant. The peripheral clocks, pancreas included, were being reset daily by nothing more than the presence or absence of food, and a compressed eating window kept every organ locked onto the same schedule. Spread eating out across 18 or 20 hours, which describes how an enormous share of people in industrialized countries actually eat, starting with coffee at 6 and ending with a snack near midnight, and the pancreatic clock is getting contradictory timing signals from every single meal. It never gets a clean enough signal to lock into a stable rhythm in the first place. This isn't only about eating late. It's about eating at inconsistent times relative to what the body is expecting, meal after meal, so the clock inside the organ never settles. Panda's group liked to compare it to jet lag. Not the kind you get from crossing time zones, but the kind an organ experiences internally, every single day, when its scheduling cues never agree with each other. Someone crossing 8 time zones feels that disorientation directly: the fatigue, the broken sleep, the foggy afternoon. A pancreas fed at 6 in the morning, again at noon, again at 4, and again at 11 at night is going through something comparable, except there's no jet lag feeling attached to it, because the organ doing the adjusting has no access to conscious experience. It can't tell you it's exhausted. It can only report, years down the line, in a blood panel, that the adjustment never fully worked. Here's the part of this that I find genuinely striking. For almost all of the time humans have existed, when to eat wasn't really a question. There was no artificial light stretching the day out, no refrigeration making food available at 2 a.m., no schedule demanding a meal because a calendar said so, rather than because the sun and the body agreed it was time. The pancreas built its clock inside that constraint, tuned to a world where food availability and daylight were the same signal. We took that constraint away in roughly a century, and the organ hasn't had time to renegotiate. It's still running the old contract. It still expects the sun to be more or less where the food is. You don't feel any of this while it's happening. There's no ache, no alert, nothing that tells you your beta cells just got handed a scheduling conflict tonight. Tomorrow morning's glucose reading might even come back normal. Nothing flagged, nothing that would show up on an annual physical for another 10 or 15 years of this same pattern repeating. That's what makes it worth sitting with. The organ isn't broken. It's doing exactly what its genetics tell it to do. It's simply being asked, night after night, to perform its most demanding task at the precise hour its own internal clock told it to power down. The mismatch is silent. And according to Marchava's mice, Shear's forced desynchrony volunteers, and Geralit's 400 dieters, it's also cumulative. So that plate at 11 at night isn't some neutral event that just gets metabolized and forgotten. It's a message sent to an organ that already knows what time it is, sent late, read anyway, and processed by machinery that would rather have been asleep.